Okay, so now we're going to look at bone structure, and bones themselves are considered organs because they are made up of different types of tissues. The main type of tissue, though, is osseous tissue, and then we also have nervous tissue, cartilage, fibrous connective tissue, muscle, and epithelial cells, which can be found within the blood vessels that are in the bone. There's 3 levels of structure. There is a gross structure, microscopic and chemical structure, and so we're going to look at that structural organization in the next few slides, or next several slides. So looking at the gross anatomy, we have both compact and spongy bone. So compact bone is going to be dense outer layer on every bone, and it appears smooth and solid. So over here on this models of the bone, that would be the outer layer. All of that outer layer that I'm pointing to there on the model below here, this outer layer would be considered compact bone. And then we have spongy bone, which is made up of a honeycomb-like or needle, a honeycomb of small needle-like flat pieces called trabeculae. Make sure you know that term. So when you look in here, if we look at this area of the bone there's your honeycomb, and if we zoom in, those little needle-like structures, flat needle-like structures, are called trabeculae. And then if we look and zoom in, this is what one would look like if we did a cross-section through it. Anyhow, the spongy bone is going to have open spaces between the trabeculae, which may be filled with red or yellow bone marrow. So now looking at short, irregular, and flat bones, these types of bones do have a similar type of structure. They're going to have spongy bone on the inside, and you can see on the right, zoom in right in that area there on the inside, you have that spongy bone, and it's going to be called diploe, and it's going to be covered with compact bone on either side. The compact bone itself will be covered with connective tissue, and on the outside of the bone will be the tissue would be called, or the membrane would be called the periosteum, and peri means to surround or cover. And then on the inside, covering the inside of the bone, would be the endosteum. Bone marrow is going to be scattered throughout the spongy bone. There will be no defined marrow cavity like there are in long bones. Hyaline cartilage will cover areas of these types of bones that are part of movable joints. For example, if you look at the short bones of the wrist, where those bones come together, there'd be articular cartilage there, and that cartilage would be hyaline cartilage on the ends. And in here we can zoom in on the picture on the right, and you can see the honeycomb-like structure, and these structures within there are the trabeculae of the spongy bone. And in the hollow spaces where you find the marrow. The structure of a typical long bone, we see the diagrams on the right. Just like with flat bones and short and irregular bones, we're going to have a periosteum and endosteum membranes, and the peri would be surrounding. As far as the bone goes, the tubular shaft that forms the long axis of the bone is called the diaphysis, and so we see that shown on the diagram on the right. And there the compact bone is going to surround a central medullary cavity. The medullary cavity is going to be where we find marrow, and in adults we find yellow marrow, mostly in that medullary cavity. On the ends of the long bones, you're going to have the epiphyses, which are widened ends of the long bones. So we can see we have the proximal on the right there, and then on the lower end of the diagram we have the distal, or part further away, epiphyses, and the ends are going to be covered with articular cartilage, which would be hyaline cartilage. Now between the diaphysis and the epiphysis, we're going to have what's called the epiphyseal line, and this epiphyseal line is made of bone, compact bone, and it's a remnant of childhood epiphyseal plate, which was made of cartilage. And in children, this is where the bones grow or lengthen. So over here in the diagram, there's your epiphyseal line right there. And if we zoom in here to a chunk right here, we can see the structure there located near the epiphyses. You can see you have your articular cartilage on the surface, there's your compact bone on the outside, and then on the inside you have your spongy bone. Looking more closely at the membranes here, There's two types. There's the periosteum and the endosteum, as what's already been mentioned. And for the periosteum, it's going to be a double layer membrane that covers the external surface, except for the joints where you have articular cartilage instead. So one of the layers, the outermost layer, is going to be the fibrous layer, and it's going to be made of dense irregular connective tissue. And if we pull it away, like you see in the diagram here, you're going to see that the periosteum itself is going to be connected to the bone matrix by these perforating fibers, also called Sharpie's fibers. So I'm going to indicate that there on the diagram. You can see the perforating fibers that go into the bone. And then we have the innermost layer is going to be called the osteogenic layer. And in that osteogenic layer, you're going to have osteogenic stem cells. And these cells are going to give rise to all the cells that we find that help develop and maintain and break down bone and so on. This periosteum is also going to contain nerve fibers and blood vessels, and those structures will enter into the bone itself through nutrient foramen. So the nutrient foramen isn't exactly labeled on this diagram here, but I'm going to go ahead and circle one over here. On the right, there's an example of a nutrient foramen. You can see a blood vessel entering in right there. And the periosteum itself, the fibrous layer, is going to be the anchoring point for tendons and ligaments. Tendons are going to be what holds muscles to bone, and those tendons pull on the bones to move them around. And then we have the ligaments, which help hold bones to bones, and those are going to be made of dense, regular connective tissue. Looking at the endosteum now, and so on the diagram here, here's your endosteum lining the medullary cavity of the diaphysis of the long bone there. There's going to be a delicate covering, delicate membrane covering the bone, that inner bone surface. It also will cover the trabeculae and spongy bone. It also lines the canals that pass through the compact bone, so there's several network of canals that go through there. And like the periosteum, the endosteum will contain these osteogenic cells that can differentiate into those other bone cells. The bones do have hemopoietic tissue, and hemopoiesis is blood formation, and so that's going to occur in the type of marrow called red marrow. And this red marrow will be found within the trabecular cavities of spongy bone and in diploi of flat bones, such as the sternum. In newborns, the medullary cavity and all spongy bone will contain red marrow. However, as development continues through childhood and into adulthood, we won't find the red marrow in there anymore. Instead, the red marrow will be located in very specific locations, which are highlighted in red in this diagram here, like the flat bones of the skull, the sternum, the ribs, the pelvis, parts of the pelvis, and the proximal ends of the femur, and the humerus will be where you find the red marrow. And the yellow marrow would be found in the medullary cavities of the other bones. That yellow marrow, though, can be converted to red marrow if a person becomes anemic. Anemic is when you have a low amount of blood cells around there, so your oxygen, not able to carry oxygen around to your body as well. So that yellow marrow can convert back to red marrow. Then we have bone markings. This is something that we've seen or you will see in the lab. These are going to be sites where muscles, ligaments, and tendons attach on external surfaces of the bone. There'll also be areas involved in joint formation or will serve as conduits for blood vessels and nerves to go through. There's three types of bone markings in general. There is projections, there are depressions, and then there are openings. The projections are basically going to be an outward bulge or bump on the bone, and these structures may be due to the increased stress from muscles pulling on the bones, or it's a modification for a joint. So sometimes these structures come about by the action of muscles on those bones. The bones will modify based on those stresses on the bone. Depressions are going to be bowl or groove-like cutout that can serve as passageways for blood vessels and nerves, or play a role in joints. And then openings are going to be holes or canals in the bone that serve as passageways for blood vessels and nerves. The next couple of slides are going to look at specific examples and very specific names for certain bone markings. These are things, again, that we've seen or will see in the lab. So this particular part of the table is going to cover projections. Okay? And the first one is just tuberosity. And so a tuberosity is going to be a large rounded projection, and it may be roughened. So if we look over here on the hip bone, on part of the ischium, there is a tuberosity right there called the ischial tuberosity. And then looking at the next projection is a crest. There's going to be a narrow ridge of bone, usually prominent. And here on the ilium, we have the iliac crest right there. So there's that crest right there. And then we have a line. Oh, I skipped trochanter. Let's go back to trochanter. Trochanters are very large, blunt, irregular shaped processes, and the only examples are going to be found on the femur. And so on the femur, we have these trochanters right here and right here. And then we have a line. So a line is going to be a narrow ridge of bone, which is less prominent than a crest. And here we have an example of a line between the two trochanters, and that's called the intertrochanteric line, inter for in between the two. And then we have a tubercle. A tubercle is going to be a small rounded projection or a process. And so over here we have an adductor tubercle. So there's your tubercle right there. And then we have epicondyles. An epicondyle is going to be a raised area above a condyle. So down here we have a condyle right there, which is a rounded area. And right above there on both sides we have epicondyles. So those are the epicondyles right here and right here. And then the spines are going to be sharp, slender, often pointed projections. Okay. So here we have the ischial spine, all part of the ischium. And then process, which is a general term for any prominence. And then over here on a vertebrae, we have projecting posteriorly from a bone in the back called the spinous process there. Now we're going to look at projections that help form joints. Okay, so the first one is head, and the head is going to be a bony expansion carried on a narrow neck. So that's actually another bone marking there. So here we have the narrowed neck right here, and then right here is the head, and this is for a rib. So the rib has a head on there. If we go back to the femur also has a head right there, the head of the femur, and then there's a neck right there. Then we have facets. And facets are going to be smooth, nearly flat articular surfaces. So right there on the head, there's a surface right there, and its smooth surface is going to be called a facet. We also have facets. Examples would be here on the vertebrae here. There's a facet right there, and right there, which help these the vertebrae articulate with either other vertebrae or the ribs that connect to some of the vertebrae in the thoracic region. And then we have condyles. I mentioned that one earlier. Those are going to be rounded articular projections. There's a condyle right there. That's called the condylar process on the mandible. And then we saw one on the previous slide. There's a condyle right there on the distal end of the femur. And then a ramus. A ramus is going to be an arm-like bar of the bone. And so if we look at the jaw here, here's your ramus, the arm-like bar of the bone of the mandible. There are other examples of ramuses, or is it rami? Right here we have there's a ramus right there and a ramus right there. They kind of fuse together from the ischium and the pubis. So we have the pubic ramus and the ischial ramus right there. And then we have depressions and openings. There's a groove, which is a furrow. And so here's your groove right here. That's on the inside of the mandible. And then a fissure is going to be a narrow slit-like opening in the orbital cavity. And here you can see the fissures in there, and they're pointing to the inferior orbital fissure. And then a foramen is going to be a rounded or oval opening through the bone. There are foramina right here, for example. Those are the mental foramen. There's a real large one on the bottom of the occipital bone where the spinal cord comes out. and it's called the foramen magnum, which means large rounded opening. And then we have a notch. A notch is going to be an indentation on the edge of a structure. For example, right here in the mandible, we have a notch between the two processes there up on the mandible. It's pointed to right here. We can go back to the hip bone over here and we can see examples of notches. There's a notch right in there. There's a notch right here. And there's a notch right there. And these notches would have specific names for them if we were to study the bone markings more closely. And then we have meatus. These are going to be other types of bone markings. A meatus is going to be a canal-like passageway. And here's an example of a meatus. This is where the ear is located. And then we have sinuses. Yeah, sinuses are going to be cavities within bones. And so here, if we cut through the skull here, we would see there's an example of a sinus right there. And then a fossa is going to be a shallow-like, basin-like depression in the bone, often serving as an articular surface or surface for connection. And so, let's see, where do we have a example here of a fossa? Well, in here we have fossa within the cranium, the cranial cavity there. But if we come back over here, now we're looking at the other side of the hip bone. So, but there are examples of fossa on there. In fact, in the lab, you've probably already seen the fossae on the scapula and on the hip bone. So let's move along now and look at the microscopic anatomy of the bone. So we're going to look at the cells of the bone tissue. There's going to be 5 major cell types, and they all specialized. They have specialized functions. There's the osteogenic cells. Those are going to be the stem cells that give rise to the other cells. Then we have osteoblasts, osteocytes, bone lining cells, and osteoclasts. So in the diagram I provided here, it's not in your textbook, but this shows where these cells come from. Your osteogenic cell is your basic stem cell, which gives rise to the bone cells. With the right signals, the osteogenic cell will transform into a osteoblast, which is responsible for building bone. And then once the bone is built around the cell, then it becomes an osteocyte. The osteoclasts are diagrammed below, and those osteoclasts actually have several nuclei. They're a larger cell, and they don't have the same origin. The osteoclasts come about by the fusing and differentiation of stem cells that come from the hematopoietic tissue or blood-forming tissue. So let's take a look at each cell individually. So here's our osteogenic cell. It's also called the osteoprogenitor cell. They are mitotically active, which means they divide by mitosis. They are stem cells, which means they can differentiate. And they are going to be found in the periosteum and endosteum that was mentioned earlier on the osteogenic layer of the periosteum and endosteum. Now, when they are stimulated, they do differentiate into osteoblasts or into bone lining cells. And some will remain as osteogenic stem cells. We have the osteoblasts. These are the bone builders or bone forming cells that secrete unmineralized bone matrix. That unmineralized bone matrix means it doesn't have the calcium in it yet. It's called osteoid tissue. The osteoid tissue is going to be made up of collagen and calcium binding proteins. So this proteins will bind or pick up calcium or calcium salts from the interstitial fluids. The collagen will make up 90% of the bone protein. Osteoblasts are active mitotically, so they can divide and make more osteoblasts. Then we have the osteocytes. These are going to be the mature bone cells, and they're going to be found in lacunae, which are the cavities. They will no longer divide. Their role is to help maintain bone matrix and act as stress and strain sensors. So they do respond to this mechanical stimuli of the increased forces or of decreased forces due to weightlessness. And so they serve a role in helping to maintain bone that would be necessary for healthy, strong bones. They also communicate information to osteoblasts and osteoclasts, which the osteoclasts are the bone breakers or bone destroyers, which they break down the bone. And then we have the bone lining cells. These are going to be flat cells on the bone surface believed to also help maintain matrix, along with the osteocytes. On the external bone surface, the lining cells are called periosteal cells, and on the internal surface they're going to be called endosteal cells, for endosteum and periosteum. And then we have the osteoclast. These are the multinucleated cells. They are derived from hematopoietic stem cells, or from the same ones that can become macrophages. They are giant multinuclear cells, and they function in what's called the breakdown of the bone, what's called resorption. Okay? And when active, the cells are located in depressions called resorption bays. And so in the diagram that I showed, that I put on this slide here, zoom in there, you can see on the bottom right there's your resorption bay right in this area right here, and there is one of the osteoclasts in that bay where it is breaking down bone tissue. So that's right down there in this area. There's your resorption bay with the osteoclast inside. The cell does have ruffled borders. You can see the ruffled border right there, which helps increase surface area for enzyme degradation of the bone. The cells also will help seal off the surrounding matrix so that we don't dissolve any bone around it. I have a diagram that'll show that in a bit on one of the next sections. And then looking at compact bone, it's again something we've seen or will see in the lab. It's also called lamellar bone because of the layers of the lamellae. It's going to consist of an osteon, which is also an older name called a Haversian system, canals and canaliculi, and interstitial and circumferential lamellae. So we look at the diagram over here, we can see some of those structures there. An individual osteon will run the length of the bone, and we'll look at it from the cut surface. The cross section is going to look like concentric layers or lamellae. Then we have the circumferential, which will go the entire length around a bone itself. And in between each individual osteon, we're going to have the interstitial lamellae within there. So let's take a closer look. So let's look at an individual osteon. We saw earlier that these osteons are going to run the length of the bone, and there are several within that compact bone sample that's in the diagram there. And it is considered a structural unit for compact bone. They are elongated cylinders that run parallel to the long axis of the bone, and they act as tiny weight-bearing pillars. And so we see a diagram of an individual one on the right. They do have several rings of bone matrix called lamellae. So lamella just means a layer. And then the collagen fibers are going to run in different directions in adjacent rings. And you can see that illustrated to the right. You can see these collagen fibers on each layer are running in different directions, and that's kind of important because that helps to withstand stresses and resist twisting from the bone. And then the bone salts are found between the collagen fibers, so the bone salts will be laid down between the collagen fibers themselves. Also notice that along the inside there is the structure called the central canal or Haversian canal, where you find blood vessels, lymphatic vessels, and the yellow structure there representing nerves, nervous tissue. So the canals and canaliculi, I pointed out the central canal already. It's going to run the length of the core of the osteon, which we were looking at earlier. It's going to contain blood vessels and nerve fibers. And then we have these perforating canals. They're also called Volkmann's canals. These are going to be canals that are lined with endosteum, and they occur at right angles to the central canal. And what they do is they're going to connect blood vessels and nerves of the periosteum, medullary cavity, and central canals. So we see them. There's an example of a Volkmann's canal or perforating canal right there. We have another one identified right here. And so overall, it provides this network of connections to those blood vessels all the way into from the nutrient form and all the way into the medullary cavity. So now looking more closely at let's look at a piece of an osteon. And so when we look in there, we're going to notice there's lacunae, which are going to be the small cavities where osteocytes occur. And then there's going to be canaliculi. which connect the lacunae to each other and to the central canal. So when we look at the diagram here, here's the central canal right here where the blood vessels and nerve fibers are. And then in the little cavities there, those canaliculi is where you're going to find your osteocytes. And then these little lines that reach out, they look like they're reaching out between the lacunae, are called canaliculi. And that's where the osteocytes communicate with each other and toward the central canal, and they're able to pass information back and forth to each other through gap junctions in their cell junctions between them. And they're also able to pass nutrients in and waste back out to the circulation. So let's move on. Now we have the interstitial lamellae. So this lamellae, these are lamellae that are not part of any specific osteon. Some fill gaps between the forming osteons, and others are going to be remnants of old osteons that have been part of the remodeling process. So when we look over here, we do see lots of lamellae. This is in the section of bone here. So if I go and I point right here between these four osteons, there's interstitial lamellae right there. And then we have circumferential lamellae, and circumference means to go around the bone. So here we have our bone laying down here, and then we come back and we look over here at a piece of that of the side of the bone, of the medullary cavity. And we look, we can see the circumferential lamellae, which are pointing to right here, and they run along the circumference of the bone. They're just to the periosteum, and they are going to be layers of lamellae that extend around the entire surface on the diaphysis, and they do help the long bone to resist twisting forces. So looking at spongy bone now, there appears to be poorly organized. But is actually organized along lines of stress to help bone resist any of the force stresses on there. So we'll tend to see structurally that the direction of the trabeculae within the spongy bone are going to follow some of the lines of force that the bones experience. The trabeculae are going to be like cables on a suspension bridge to help provide strength to the bone. They do not have osteons, but the trabeculae do contain irregularly arranged lamellae and osteocytes interconnected by canaliculi. So here, if we take a look, here we have, let's say this is a little portion of the diaphysis that we have right here, and then we zoom in right here, and then we look at some of the spongy bone found towards the inside, and then we zoom in on this structure there. There you see your trabeculae there, and we section through the trabeculae, and you can see the lamellae. And so we zoom in right here, and there you can see the irregularly arranged lamellae there. There is no central canal, there is no osteon. And the reason for that is that the endosteum on the outside does have blood vessels, and those provide enough exchange for the very few layers that are there. So the trabeculae are quite thin, so there's enough blood supply to allow these cells within the very few layers of the lamellae to be able to pass nutrients and waste products back and forth, and for them to communicate with each other there. So there's no need for a central canal. So bone, looking at the chemical composition, bone is made up of both organic and inorganic components. The organic components include the bone cells that we find there: osteogenic, osteoblast, osteocytes, lining cells, osteoclasts, and also the osteoid. The osteoid is going to make up 1/3 of the organic bone matrix, and it is secreted by the osteoblasts. It does consist of a ground substance, the osteoid does, and collagen fibers, which contribute to the high tensile strength and flexibility of the bone. The resilience of the bone is due to sacrificial bonds in or between the collagen molecules that stretch and break to dissipate the energy and prevent fractures. So when the bones are experiencing stress, some of the collagen fibers will break and help relieve some of that energy. Now, if no additional forces or trauma is on there, then those bonds can reform. And then the inorganic components include the hydroxyapatites, which are the mineral salts. One of the major ones is going to be calcium phosphates. and they make up 65% of the bone mass. And they consist only of tiny calcium phosphate crystals in and around the collagen fibers. These inorganic component here, these mineral salts, are going to be responsible for giving the bone hardness and resist to compression. So that's the role that the inorganic salts form. So really the properties of the bone being both flexible and resistant to compression is due to the combination of the properties of both the collagen fibers, or the organic components, along with the inorganic components. The bone is half as strong as steel in resisting compression, and as strong as steel in resisting tension. So this inorganic component does last long after death because of mineral composition.